Hello students, this is your Prasanna ma'am. Today I am here to deal with the most crucial topic, that is DNA replication, transcription and translation from the chapter Molecular Basis of Inheritance. So, if you look into the central dogma of molecular biology says that DNA gets replicated and the information from DNA is transferred to RNA. This is called as transcription, and information in RNA is coded in the form of proteins. This is translation. So, what is the ultimate goal of DNA beta? The ultimate goal of DNA is to synthesize proteins. So, to conduct DNA replication, what is the major enzyme that is used to conduct replication? DNA dependent DNA polymerase is used. DNA dependent DNA polymerase is used. And to conduct transcription, where RNA is being synthesized from DNA, here the enzyme used is DNA dependent RNA polymerase. DNA dependent RNA polymerase, wherein RNA strand synthesis is dependent on DNA strand. We are synthesizing RNA strand, so you are calling RNA polymerase. Synthesis is dependent on DNA strand, so this enzyme is DNA dependent RNA polymerase. In few organisms like retroviruses, where HIV, the genetic material is RNA, but to conduct this process, RNA need to be converted or transferred to DNA. In that case, the enzyme used is RNA dependent DNA polymerase, RNA dependent DNA polymerase, wherein DNA strand is synthesized from RNA. What is synthesized ma? DNA strand is synthesized from RNA. You will call it as DNA dependent RNA polymerase. This enzyme is also called as reverse transcriptase. What is it? It also called as reverse transcriptase, and the process is called as reverse transcription, and ultimately the information from RNA is synthesized to proteins. If you take in prokaryotes, the site of replication, transcription and translation, both in prokaryotes and eukaryotes, if you see prokaryotes and eukaryotes. In prokaryotes, as there is no membrane bound nucleus, what happens is the DNA is in the form of nucleoid. It is in the form of what beta? It is in the form of nucleoid, which is non-membrane bound. This is non-membrane bound nucleoid, inside which DNA replication happens. Inside which what happens? DNA replication happens. If you take in the cytoplasm, both the transcription and translation are coupled. Transcription and translation are coupled in the cytoplasm, because by the time of mRNA synthesis, translation begins directly. By the time of mRNA synthesis, translation process begins in prokaryotes. Process begins in prokaryotes. So, these two, transcription and translation, both takes place in the cytoplasm. It takes place in where beta? Cytoplasm of prokaryote. Both transcription and translation both takes place in where? In the cytoplasm of prokaryotes. If you take eukaryotes, wherein eukaryote has membrane bound nucleus. Eukaryotes have what ma? Membrane bound nucleus. Here the genetic material whichever is there, inside the nucleus only DNA replication happens. Inside nucleus what happens ma? DNA replication. And mRNA synthesis also happens inside nucleus. Both transcription, post-transcriptional changes occur in eukaryotes. Post-transcriptional changes also take place inside the nucleus. Post-transcription also takes place inside the nucleus. In cytoplasm, synthesis of proteins, that is translation, post-translational changes occur inside where? Inside the cytoplasm of a eukaryotic cell. So, clear with the sites or locations of both replication, transcription and translation, and then you are clear with the enzymes of DNA replication, transcription and reverse transcription as well. Now, if we look into the replication keenly, what method of DNA replication is widely accepted and proved? The widely accepted method of DNA replication is DNA replication that is semi-conservative method of DNA replication. What is that method, ma? Semi-conservative method of DNA replication. Semi-conservative method of DNA replication, which means that from a double-stranded DNA, each strand acts as template. From a double-stranded DNA, each strand acts as template. Each strand acts as template or parent strand. Each strand acts as what, ma? Template or parent strand. From each template or parent strand, new strand is synthesized. New strand is formed in complementarity with the template or parent strand. From each strand of DNA, new strand is formed where each strand of DNA will act as template or parent strand. This is the semi-conservative method of DNA, which is widely accepted and proved by Michelson and Stahl's experiment. Now, if we look into the enzymatic machinery of DNA replication, what is that, ma? Enzymatic machinery of DNA replication. Enzymatic machinery of DNA replication, wherein, as we already learned, that the main enzyme which conducts the process of replication is what, beta? DNA dependent DNA polymerase. DNA dependent DNA polymerase. But then there are steps to conduct the replication process. The very first step is to identify origin of replication site. So, inside a prokaryotic cell which has circular, mostly it has what type of DNA, ma? Circular double-stranded DNA. It has circular double-stranded DNA. Inside a prokaryotic cell, you have only one origin of replication. Inside a prokaryotic cell, you have only one origin of replication, which is said to be ORIC. But if you take a linear double-stranded DNA from a eukaryotic cell, linear double-stranded DNA from 5 prime to 3 prime, 3 prime to 5 prime. There are multiple ORI sites in a eukaryotic DNA. There are multiple ORI sites. In a eukaryotic DNA. In a prokaryotic, you have only one ORI site. In a eukaryotic DNA, you have multiple ORI site, origin of replication sites in a eukaryotic DNA. Once after identifying the origin of replication region, the very next step need to be conducted is activation of deoxyribonucleotides, activation of deoxyribonucleotides. Why this activation of deoxyribonucleotides is required? It is required because the enzymes like helicase, topoisomerase to conduct the process of replication, they are all energy driven and the energy needs to be supplemented from these nucleotides. So, deoxyribomonophosphate like deoxyribothymin monophosphate, deoxyribocytidine monophosphate, deoxyriboadenine monophosphate and deoxyriboguanine monophosphate. These are added with a phosphate group. These are added with what ma? Phosphate group by the enzyme phosphorylase, by the enzyme phosphorylase. And then they are converted to energy rich deoxyribothymin triphosphate, deoxyribocytidine triphosphate, deoxyriboadenine triphosphate, deoxyriboguanine triphosphate. Okay, all these are like ATP only, they are energy rich. Now, the energy whichever is required for the enzymes to conduct the process of replication are supplemented by these triphosphates. Is this clear beta? So, what is the first step of replication? To identify the ORI site. What is the second step? Activation of the nucleotides. So, what happens is once the activation is done, once the origin of replication site is decided or identified, the very first enzyme that comes and cleaves the double stranded DNA is helicase. Helicase is ATP dependent. Helicase is what ma? ATP dependent. This helicase comes and it will cleave the two strands of DNA and gives an appearance of replication fork. As the two strands immediately after cleaving, they are single stranded, right? So, they get unstable. To maintain the stability of single stranded DNA, single stranded binding proteins will come and attach to those single stranded DNAs. What happens ma? Single stranded binding proteins will come and attach to the single stranded DNAs whichever are cleaved by helicase. The next, as the double stranded DNA gets cleaved by the helicase, on the other side torsions occur. What happens ma? Torsions and supercoils are seen on the other side of the DNA. So, these are set properly by topoisomerases. These are set properly by topoisomerases. Even they are ATP dependent. Even they are what ma? ATP dependent. Now, from this region at the start site to start or initiate the process of DNA replication, firstly, the primase enzyme comes and it will synthesize RNA primer in the opposite strand of the DNA. Always do remember, either in the synthesis of DNA or in the synthesis of RNA, synthesis always takes place from 5' to 3' direction. Synthesis always occurs from 5' to 3' direction, identifying what ma? Identifying the OH group which is present at the 3' end of the template strand. Identifying OH group from 3' end of template strand, from 5' to 3' the synthesis occurs. So, firstly what is added? RNA primer is the starter of the DNA replication. RNA primer, it acts like what ma? It acts like starter of DNA replication. It acts like starter of DNA replication, where it initiates or begins the process of DNA synthesis. Till here it is clear now? Next if you see, from here you will clearly understand how a new DNA strand is synthesized. So, from the parent strand, whichever replication fork, if you see replication fork which is formed from the parent strand, just a minute beta. So, from here which is 5' to 3', 3' to 5'. So, here the DNA synthesis towards the replication fork occur from 5 prime to 3 prime. RNA primer starts the reaction. What starts ma? RNA primer acts like starter for the DNA synthesis. Now once RNA primer is synthesized, from here the DNA is synthesized by the enzyme DNA dependent DNA polymerase. The most effective enzyme which highly activates the DNA synthesis is DNA polymerase 3. What is that enzyme? DNA polymerase 3 in prokaryotes, wherein DNA polymerase 3, it is that accurate wherein it adds around 2000 base pairs per second. How much accurate beta? It adds around 2000 base pairs per second. So this synthesis takes place towards the replication fork. The strand whichever is synthesized towards the replication fork is called as leading strand. It is called as what ma? Leading strand. And the strand which is synthesized away from the replication fork, as it is away from the replication fork, there is not just one RNA primer, there are many RNA primers required to synthesize a DNA strand away from the replication fork. So as there are many RNA primers, these RNA primers are called as Okazaki fragments. They are called as what ma? Okazaki fragments. These Okazaki fragments are combined, these Okazaki fragments are continued with DNA synthesis. Once a DNA gets synthesized totally, they are ligated, they are attached by DNA ligase. They are attached by what beta? DNA ligase. So if you look into it properly, firstly from 5 prime to 3 prime and 3 prime to 5 prime. So the strand which is synthesized towards the replication fork is called as what ma? Leading strand. It is called as leading strand. Just a second. Here it is 3 prime, here it is from 5 prime to 3 prime. This is 3 prime to 5 prime. Here again it is 3 and 5. Yes. So yes, it is synthesized from 5 prime to 3 prime, identifying the OH group at 3 prime region, wherein the synthesis is from 5 prime to 3 prime and it is towards the replication fork. This is called as what ma? Leading strand. It requires only one RNA primer. It requires only one RNA primer. Whereas here if you see from 5 prime to 3 prime, away from the replication fork, logging strand is synthesized, wherein it requires multiple RNA primers. It requires multiple RNA primers, wherein it is called as Okazaki fragments. They are called as what ma? Okazaki fragments. And once the DNA polymerase synthesizes DNA over it, those DNA fragments need to be combined together, attached together by the enzyme DNA ligase. By the enzyme what beta? DNA ligase. So here if you look into all the enzymes which are involved in the replication, the very first enzymes which come to the picture is helicase. Helicase, identifying the ORIC, and after activation of deoxyribonucleotides, this helicase will do what beta? They will cleave the double stranded DNA, and the helicase is ATP dependent. It is ATP dependent. And the second enzyme is single stranded binding proteins. Second proteins are what? Single stranded binding proteins which maintain the stability of the single strands. They will maintain what ma? Maintain the stability of single strands of the DNA whichever are cleaved by the helicase. And third enzyme which comes to the picture is topoisomerases. Topoisomerases. These topoisomerases are also ATP dependent. They are also what beta? They are ATP dependent. They will clear the supercoils or the torsions which are created on the other side of the DNA because of the helicase activity. Topoisomerases in prokaryotes, they are known as DNA gyrases. They are known as what ma? DNA gyrases. If you look into the fourth enzyme, that is primase. What is that ma? Primase that adds RNA primer, which acts like a starter for the DNA synthesis. RNA primer is added at what place of the template strand ma? Identifies the 3' OH group. The synthesis is always started from 5' to 3'. RNA primer is added at this region. Next, fifth enzyme is DNA polymerase. What is that beta? DNA polymerase. DNA polymerase, wherein DNA polymerase will synthesize the DNA strand most effectively, like 2000 base pairs per second. And sixth enzyme is DNA ligase. What is that beta? DNA ligase. Even DNA ligase is ATP dependent. It is also ATP dependent. DNA ligase will add, or it will combine all the DNA fragments in the lagging strand of the DNA, and it will synthesize the new strand away from the replication fork. So, I hope all the enzymes for DNA replication and the process of DNA replication is clear to you. Now, the process of DNA replication is done. We are moving to the transcription of DNA to RNA. Transcription of DNA to RNA. So, after just a second. Yeah. So, here if you see leading strand, now you have to tell me here what is the leading strand and what is the lagging strand. This is the leading strand which is continuous. This is what beta? Leading strand which is continuous towards the replication fork, and this is the lagging strand which is discontinuous away from the replication fork. Lagging strand. So, if you look into here, both the leading strand and lagging strand, they both make one more double stranded DNA. From this double stranded DNA, only a part of the DNA will help in the synthesis of protein, that is known as cistron. Only a part of the DNA that is helpful in the synthesis of protein, where you will call it as what ma? Cistron. So, if you take as it is these double stranded DNA for transcription, transcriptional unit includes promoter, second is structural gene, and third is terminator. Third is what ma? Terminator. So, if you look into this double stranded DNA, of which one strand is called as template strand and the other strand is called as what ma? Coding strand. One strand is called as template strand and the other strand is called as coding strand. Template strand is also called as sense strand. It is also called as sense strand, as RNA strand whichever is synthesized is complementary to the template strand. RNA strand, mRNA whichever is synthesized, it is complementary to mRNA is complementary to the template strand of the DNA. Template strand. And the codons whichever are present in the mRNA are similar to the coding strand except uracil in the place of thymine. And coding strand, it is also called as antisense strand. It is also called as antisense strand or non-template strand also. So, here at the 5' region of the coding strand, Promoter is situated at upstream. You will call it as what ma? Upstream. Where is upstream present? If the question comes, it is present at the 5' region of coding strand. And where is terminator present? It is present at the 3' region of the coding strand and 5' of the template. This is least bothered, but most important is you need to remember upstream or the promoter region is present at the upstream, that is at the 5' region of the coding strand, and terminator is present at the 3' region of the coding strand. If coding is placed here and template strand is placed over here, then the definition will reverse. If here it is the coding strand, then you tell me what is promoter. This is the promoter and this is the terminator. Clear beta. So where is promoter present? Fix it in your mind. It is present at the 5' region of the coding strand. Clear? Is this fine? Transcriptional unit includes 3 components. What are they? Promoter, terminator and a structural gene. Promoter is towards the upstream, terminator is towards the downstream. Clear beta? Now if we look into the transcription or the gene, before entering into the transcription in detail, let us understand what is gene in specific. Okay, what is a gene? Gene is nothing but a functional unit of inheritance. We will call gene as what ma? Functional unit of inheritance. Gene is called as functional unit of inheritance. Means whichever fragment from the DNA is coding for a protein, whichever fragment from the DNA or a unit from the DNA is coding for protein is called as gene. So if you take in a eukaryote, from the promoter region and terminator region, in between whichever structural gene is there, this structural gene will synthesize only one protein. This will code for only one protein, one polypeptide chain, and it will synthesize or process only one polypeptide chain. That is why this is called as monocistronic. That is why this is called as monocistronic. If you look into here in a prokaryotic cell, from promoter region, in between promoter region and terminator region, there are multi-cistrons. Many polypeptide chains are formed. This is because mRNA is flanked by many ribosomes. mRNA is flanked by many ribosomes, which is said to be polysome or polyribosomal condition, wherein this type of polysome will result in the synthesis of multi-chain, or process polypeptide chain, multiple proteins, that is polypeptide chain. Okay, happens only in the prokaryotes. That is why in a eukaryotic mRNA, from a eukaryotic mRNA, only one polypeptide chain is synthesized. That is why this is called as monocistron. Okay. And what is happening in the prokaryotes? In prokaryotes, many protein chains are formed, many polypeptide chains are formed which are different. That is why they are called as polycistron. They are called as what ma? Polycistron. So I hope this is clear. Monocistronic and polycistronic formation of polypeptides, and polycistronic polypeptide chain is formed as a result of polysomic condition, which is seen only in the prokaryotes. And what is cistron beta? Cistron is nothing but the protein chain which is expressed from a fragment of DNA or a gene. Protein chain which is formed, or a polypeptide chain which is formed from the fragment of gene, that is cistron. What is a gene? Functional unit of inheritance or functional unit of DNA strand is called as what ma? Gene. Clear? Now, if we look into the details of transcription, first we will see what is a prokaryotic transcription. In a prokaryotic transcription, three phases are present, and only one enzyme that governs this process is RNA polymerase. What RNA polymerase ma? DNA dependent RNA polymerase. What RNA polymerase beta? DNA dependent RNA polymerase, and it is divided into 3 stages. One is initiation, second is elongation, and third is termination. At the time of initiation, what happens is RNA polymerase along with sigma factor will come and attach to the transcriptional unit. It will come and attach to what beta? Transcriptional unit, that is promoter region. It will come and attach to what region ma? Promoter region. One extra point to be dealt beta, before the promoter region and after the terminator region. Before the promoter region and after the terminator region, there are few regulatory sequences which are called as untranslated regions. What are they called as? UTR, that is untranslated regions. And before the promoter, that is on the upstream, after the terminator, there are few more sequences present called as regulatory sequences. Regulatory sequences which regulate the process of both transcription and translation. Keep these both things in your mind. And coming back to the initiation concept, if you look here, sigma factor is binding along with RNA polymerase, forming what beta? Holoenzyme. This RNA polymerase binding along with sigma factor forms holoenzyme. This enzyme will come and attach or bind over which region? Promoter region. This forms the initiation. In elongation process, transcription bubble is formed. What is formed dear? Transcriptional bubble. What is formed beta? Transcriptional bubble. Here transcriptional bubble is formed, and this RNA polymerase will synthesize the DNA, will synthesize the RNA from DNA, again from which strand to which strand? From 5 prime to 3 prime. Okay, it is synthesizing from 5 prime to 3 prime. And in a prokaryotic cell, where does transcription happen? Transcription happens in cytoplasm. Transcription occurs in where beta? Cytoplasm. And this mRNA strand is released. By the time it is totally released, only translation process is started. That is why here in a prokaryotic cell we will say both transcription and translation, are coupled. Transcription and translation are coupled. Now, once the mRNA strand is synthesized, in termination time what happens is Rho factor will come and attach to the RNA polymerase, and it will pull the RNA polymerase into the cytoplasm, and it will release the mRNA strand into the cytoplasm. What happens ma? Here Rho factor along with RNA polymerase, it will pull the RNA polymerase and it will release the mRNA into the cytoplasm. So these are the 3 important steps which happen in the transcription, that is prokaryotic transcription. One, initiation step, RNA polymerase is binding with sigma factor in the promoter region. In elongation step, as a result, transcription bubble is formed and mRNA synthesis happens from 5 prime to 3 prime direction, and mRNA is complementary to template strand of the DNA, and it is similar to the coding strand, except in the place of thymine in mRNA uracil will be present. Third is what ma? Termination, wherein RNA polymerase along with Rho factor is pulled down into the cytoplasm and the free mRNA is released into the cytoplasm. This is all about prokaryotic transcription. If you look into eukaryotic transcription, it is similar to prokaryotic transcription, but post-transcriptional changes happen in a eukaryote and site of transcription is different. 2 things vary beta. What is site of transcription and post-transcriptional changes? In eukaryotic transcription, what happens? Firstly, Same RNA polymerase along with sigma factor will bind at promoter region. It forms transcriptional bubble. mRNA will be synthesized. Here the mRNA is called as heteronuclear RNA. It is called as what ma? Heteronuclear RNA. And in prokaryotic transcription, only RNA polymerase is involved. DNA dependent RNA polymerase we are calling it as. Whereas in eukaryotic transcription, if you see, there are 3 RNA polymerases involved. What are they ma? RNA polymerase 1, RNA polymerase 2, RNA polymerase 2 and RNA polymerase 3. RNA polymerase 3. If you look into RNA polymerase 1, RNA polymerase 1 will synthesize rRNA, that is ribosomal RNA, and important protein subunits like 28S rRNA, 18S rRNA, next 5.8S RNA subunits, all these are synthesized from RNA polymerase 1. And RNA polymerase 2 will synthesize heteronuclear RNA. RNA polymerase 2 will synthesize what beta? Heteronuclear RNA. RNA polymerase 3 will synthesize snRNA. RNA polymerase 3 will synthesize snRNA and SNRPs which are included in the splicing of hnRNA, and they are also involved in the synthesis of 5S RNA. They are also involved in the synthesis of 5S RNA. This is very, very important. And if you look into the types of RNA, be it rRNA, hnRNA, 5S RNA, or else you know rRNA, hnRNA, 5S RNA, RNA polymerase 3 is also involved in the synthesis of tRNA. All about types of RNA I have dealt in the RNA world and types of RNA session. Please look into it. Now here 3 important enzymes are involved in what? Eukaryotic transcription. In prokaryotes though only one enzyme, that is RNA polymerase. So here if you see for hnRNA synthesis, what is involved ma? For hnRNA synthesis, RNA polymerase 2 is involved. Once the hnRNA is synthesized, 3 important post-transcriptional steps happen. What happen? 3 important post-transcriptional steps. One is 5' capping. One is what ma? 5' capping. Second is splicing. Second is RNA splicing. And third is formation of polyadenylation tail. What happens? Why there is a requirement of 5' capping and 3' polyadenylation? We will do first, then we will discuss about RNA splicing. First, 5' capping, what is added over here? 5-methyl guanosine. What is added beta? 5-methyl guanosine. Triphosphate, 5-methyl guanosine triphosphate is added at the 5' region, so that during the process of translation the smaller subunit of rRNA will come and bind at the initiator codon. Smaller subunit of rRNA will come and bind at the initiator codon and initiate the process of translation. That is the reason at 5' region 5-methyl guanosine is added. If you look into 3' polyadenylation, in 3' polyadenylation, multiple adenine residues around 200 to 300 adenine residues, around 200 to 300 adenine residues are added over 3' region to ease the process of termination during eukaryotic translation. Clear? And now if we look into RNA splicing, in hnRNA you have 2 sequences, both introns and exons. Introns and exons. So here what is intron and what is exon? Exon, do remember, expressed sequences. Exon means what ma? expressed sequence. Expressed sequence. Intron means inactive regions. Intron means what na? Inactive sequence. You can remember intron inactive, exon expressed, as such you remember. Okay. So here intron exon pattern of arrangement is also called as split gene arrangement. It is also called as what ma? Split gene arrangement. It is also called as split gene arrangement, and this intron exon pattern of mRNA chain is observed in, or this intron exon pattern of sequence is observed in archaebacteria which are primitive monorans. These are observed in what ma? Archaebacteria which are primitive monorans, and then you will again see it in eukaryotes. You will nowhere observe it in a prokaryotic cell, in a bacterial cell. Okay. So this heteronuclear RNA will also, it is also acting like reminiscent of antiquity. Reminiscent of antiquity, wherein it is reminding about the ancestral cell like archaebacteria, wherein same split gene arrangement is seen. And second point is to cleave the inactive sequences and to join all the expressed sequences. This expressed sequence joining and removal of intron groups, which is said to be splicing, is reflecting the RNA dominance, RNA world dominance. The dominance of RNA world is shown here. DNA doesn't have any such feasibility to cleave its unexpressed sequences and only maintaining the expressed sequences, whereas RNA has that feasibility wherein it is removing all the inactive sequences and joining only the active sequences that are expressed sequences, which is reflecting the dominance of RNA world. These 3 points are very, very important beta, do remember in your mind. So what is released into the cytoplasm? All these post-transcriptional changes are also happening inside the nucleus. The mRNA, matured RNA chain, the matured RNA chain, mRNA chain with 5' capping and 3' polyadenylation, 5' capping and 3' polyadenylation. This sequence is released into the cytoplasm for the process of translation. I hope eukaryotic transcription is clear to you. Now we will enter into translation. If you look into translation, just a second beta, yes, translation first is what ma? Yes, translation. Now for translation we require 3 types of RNA. One is mRNA. If it is a eukaryotic RNA, we have that mRNA which is released into the cytoplasm. What is that? 5' cap, methyl guanosine triphosphate is present over here, and 3' polyadenylation chain, and only exon sequences, This is released into the cytoplasm. mRNA is done. Second is rRNA is required, that is ribosomal RNA. What is that beta? Ribosomal RNA with a smaller subunit and a larger subunit, wherein it has the protein subunits like 23S RNA in prokaryotes, 28S RNA in eukaryotes, 28S RNA in eukaryotes, and 5S common both in prokaryotes and eukaryotes. You have 18S in eukaryotes, 16S in prokaryotes, but these two I have specified because they are involved in the enzyme synthesis, that is peptidyl transferase. What is that ma? Peptidyl transferase, which is involved in the formation of peptide bond. This is involved in the formation of what dear? Peptide bond. Clear? This is involved in the formation of peptide bond, and this helps in the formation of a polypeptide chain. This is rRNA. Third is tRNA we require. tRNA. I have dealt clearly about tRNA in our previous session. Please go through it better. First, for translation process, two activities need to be done. One is activation of amino acid. One is what ma? Activation of amino acid. Activation of amino acid, wherein amino acid along with the energy rich molecule, that is ATP, is combined in the presence of amino acyl tRNA synthetase, and it forms a complex called as amino acyl AMP complex. It forms a complex, what is it beta? Amino acyl AMP complex, and inorganic phosphates are released. This amino acyl AMP complex is called as activated amino acid. After the amino acid getting activated, this amino acid is attached to the 3' end of tRNA. Where is amino acid added beta? To the 3' end of tRNA. If you look over here, this is the secondary structure or the active structure of tRNA, which conducts the process of translation, wherein at the 3' OH group, here activated amino acid is added, and here it is 5' end. Okay. In the opposite side, it has anti-codon, which codes for the sequences whichever are present in the mRNA. And here closer to the 3' end, you have T-Si-U loop, that is ribosomal binding loop, and closer to the 5' end you have DHU loop. DHU loop is amino acyl synthetase binding loop. So here over here the activated amino acid is binded and it forms charged tRNA. Second step is what ma? Charging of tRNA. Second step is charging of tRNA. Charging of tRNA, wherein at one end the charged, at 3' end the charged amino acid is added. Now this charged tRNA will come and bind to the promoter region or the beginning region, initiator codon of the mRNA. If you look here clearly, yes, this is the 5' end and this is the 3' end. This here also initiation, elongation, termination, 3 steps will be there. In translation, initiation, first is what ma? Initiation. Second is elongation, and third is termination. 3 steps are present over here too. Okay. So if you look into the initiation process, what happens is the smaller subunit recognizes the promoter region. The smaller subunit recognizes the initiator codon region, that is 5' methyl guanosine is there, right? Over here the smaller subunit of ribosome comes and attaches, and the larger subunit, larger ribosomal subunit will come and fit over here. This is the first thing which happens. Second is tRNA charged. Charged tRNA will come and sit on the A site of the, it will come and sit on the A site of the larger subunit. Larger subunit has 3 sites, A, P and E site. It has 3 sites. What are they ma? A, P and E sites. First tRNA whichever is charged will come and land on the A site, then it will move to the P site. In P site it will copy or it will code the codon whichever is there. For example, the starter codon, that is AUG, which codes for methionine. This is copied by the tRNA, and from the amino acyl synthetase loop, amino acid is synthesized. From the amino acyl synthetase loop, just give me a minute, I will draw the secondary structure of tRNA. Yes. So if you look over here, at 3' end amino acid is attached. This is the charged tRNA. This copies the AUG codon, and from the amino acyl synthetase loop it will synthesize the amino acid, for example methionine. It will synthesize the amino acid, for example methionine. Once it is synthesized, then the tRNA will move to exit side and it will go away from the ribosomal larger subunit, it will move away. Then the ribosome moves to the next tRNA, again a charged tRNA will come, land over the A site, then from P site it will synthesize one more amino acid. These two amino acids are connected by the enzyme peptidyl transferase. These two amino acids are connected by the enzyme peptidyl transferase, which is synthesized or which is released by 23S RNA subunit from prokaryotic ribosome or 28S RNA subunit from eukaryotic ribosome. As such, peptidyl transferase will connect the amino acid and this forms a polypeptide chain or protein. So, in initiation step, what is happening? The smaller subunit of ribosome will come and attach to the initiator codon over which the larger subunit fits, and charged tRNA will come and land over here. In elongation region, what is happening? The charged tRNA will copy the codon and from the amino acyl synthetase loop it is synthesizing amino acids, wherein two amino acids are connected by the enzyme peptidyl transferase. In termination region, what is happening beta? In termination region, release factor will come and attach to stop codons whichever are present in the mRNA. Release codons will come and attach to the stop codons which are present in the mRNA. Now you tell me what are the stop codons we have? UGA, UAG and UGG. Okay, these are what ma? Stop codons. UAA, UAG and UGA, these three are stop codons. Once release factor binds to the stop codon, then the translation process stops, both the ribosomal subunits will move apart and polypeptide chain is released into the cytoplasm. These are the three steps of translation. I hope the process of replication, transcription and translation is clear to you. Coming up with a very nice topic in the next class. Stay tuned. Thank you.